Hey guys, welcome and welcome back to my channel. This is Dr. Prasanjit Das, currently JR1 in the Department of Pharmacology, AIMS Kalyani, and I am back with one of my presentations. So, without wasting any time further, let's get into today's video. So guys, today I am going to present the topic that is chromatography, which is one of the separation techniques, that is analytical techniques, which is commonly used in laboratories for analyzing different samples. So in this presentation, we are going to discuss this topic under the following headings, that is the introduction to the chromatography and the history of development of chromatography, basic principles involved in chromatography, types of chromatography, and several terminologies which are related to chromatography. Following that, we will be discussing about the gas chromatography in detail, and then we have liquid chromatography, under which we are going to give a special emphasis on HPLC, that is the high performance liquid chromatography. Apart from that, we are going to discuss about the other chromatographic methods, that is TLC, that is thin layer chromatography, and SFC, that is supercritical fluid chromatography. Following that, we will be discussing about the applications and finally the summary slide. Coming to the introduction and history, so chromatography is one of the most common methods for achieving the separation of a target compound from other chemicals in the sample. It is very simple, that is, it is helpful, it is helping us to achieve the separation, separation of a target compound, that is the desired compound which we are looking for, from other chemicals in a mixture, that is in a sample. So the components of a mixture are separated based on their differential interactions with the two chemical or the physical phases which are involved in chromatography, and that is the mobile phase and the stationary phase. So I will be discussing about this mobile phase and the stationary phase in the upcoming slides. Right now, what we need to know is the separation occurs because of the differential interactions of all the different chemicals which are present in a particular sample with the mobile phase and the stationary phase. And chromatography was first used, that is, it was first, the application of chromatography was done by this gentleman, that is Michael Sweet. So Michael Sweet was the individual who has first described chromatography, that is in the year of 1903, and he used this technique for separating the several plant pigments into different colored bands, and for this he used the type of chromatography which is known as column chromatography. We will be discussing this also. So he was the person who coined the term chromatography. And why do we say this method as chromatography? This is because it is obtained from the combination of two Greek words, that is chroma and graphene. So chroma means color and graphene means to write, and that is how this name chromatography was given to this separation method. But right now, in the modern days, when we are doing chromatography, the chromatographic methods are not only used to separate the pigmented materials, but it can also separate those materials which are not having color. So right now we can say chromatography is a misnomer which is used nowadays, but this term was coined by him based on the separation of the plant pigments which we did that time, and those were colored bands. So as a result, he gave it a name, that is chromatography. Now coming to the next slide, that is the basic principles, the types of chromatography based on different aspects and the terminologies. So first, let's discuss about the basic principles. So as I've already mentioned it, the separation of the chemicals occur based on the differential interaction between the stationary phase and the mobile phase. So what is the stationary phase and what is the mobile phase? If you can see the diagram here on the right side, it is a depiction of paper chromatography. So it is a type of chromatography, it is a type of planar chromatography. So I will be discussing that later. So here you can see we have a paper. Okay, so this paper is the one which remains static throughout the entire process of the chromatographic separation. Okay, that is the separation of components. So this is what we call the stationary phase. And then you can see we have the solvent here. Okay, and here we have applied the sample, that is, it is a mixture of several components, and we want to separate the components. So as you can see here, we are having the solvent. So this solvent is going to move upwards, okay, and with the help of the capillary action, and that's why this solvent is nothing but the mobile phase, because it is moving. Okay, it is moving throughout the process of chromatography, and this is the movement of the mobile phase which is going to carry the different components to different heights in the paper. As you can see, the paper here is the stationary phase. Okay, and stationary phase, so here you can see the separated components. As you can see, it is indicated by different dots, that is red, yellow, orange, blue, and area where the sample was applied earlier. This is because the sample which you are seeing here is having different components, out of which the one which is represented by blue dot is the one which is having the maximum interaction with the stationary phase, that is the paper, and the red one is the one which is representing the least interaction with the stationary phase, and as a result it has been carried away with the solvent for a longer distance. So that is how the components are being separated. So it is mentioned here, the mobile phase travels through the system and carries the sample components with it once the sample has been applied or injected. So sample here, it is the sample that is applied in the form of a drop on this line. So the stationary phase is held within the system by a support. So support is nothing but the material which is holding the stationary phase. So here we are having a paper. So paper itself is the support and it is also the stationary phase. But we are having examples like column chromatography, where we are having a support and where we are having a stationary phase. Both will be different. So a support can act as the stationary phase directly, or else it can have a stationary phase which is coated over it. In case of the chromatography which was done by Michael Smith, he used column chromatography, and there the support which he used was calcium carbonate, and this calcium carbonate was only the support and it was also the stationary phase there. So there was no such coating of stationary phase over the lining of the column in which Michael Smith did the separation of the pigments of the plant. So this is about the different parts of the chromatography. That is the basic part, that is the mobile phase and the stationary phase and the support. So the difference in the rate of travel for the components and the separation occurs as they move through the chromatographic system, and this you can see here as a different bands. So it will be different in different types of chromatography. Here you can see in the form of different dots. In case of the paper chromatography, it is basically blotting type of pattern, but in case of column chromatography you can see some bands in. Then you can also see different there are different types of chromatography where you will get the pattern as different. Okay, and this will be interpreted later. If we are connecting it, the chromatographic system, with a detector, we can detect it in the form of peaks. So I will be discussing that later. Now let's discuss about the general components of a chromatographic system. As I already mentioned, this is an example of a column chromatography. So here you can see this is an entire column. So here the column is packed with several particles. So what do we say this column? This column is actually a packed column. Okay, so packed column means a column whose lumen is packed with the support material. So this support material will have either the stationary phase coated over it, or this support can itself act as the stationary phase, as it is shown here. So we will inject the mobile phase from above, and then with the action of gravity, the mobile phase will be going downwards and it will interact with the stationary phase. And here you can see the first band, that is B. This means this component of the sample is having more interaction with the stationary phase. That's why it is being formed in the earlier part, and the later part you can see another component, that is A, which is representing another band. So here it is the component which is having less interaction with the stationary phase, and hence it is moved with the mobile phase for a longer distance. And then all these components, it will take some time to come out of this column chromatograph, and it will be detected with the help of a detector which is present here. It will be connected to the detector, and we can see the different components as different peaks. Okay, so here this peak is representing the A band, that is the A component. When it comes out, the process of it coming out of the chromatographic system is what we call as elution. Okay, following that we are getting B, because B is upwards. So this is basically the component of the chromatography system, and that is the chromatograph. Here we have described about the column chromatograph. Now coming to the classification of chromatography. So first, based on the mobile phase which is used in chromatography, we can classify the chromatography into different types. First is gas chromatography. So from the name it is clear, here the mobile phase is gas. Then next is liquid chromatography. Here the mobile phase is liquid. Then we have supercritical fluid chromatography. Here the mobile phase is supercritical fluid. So what is the supercritical fluid? Supercritical fluid is the fluid which is having the property of both gas and liquid. So we can use carbon dioxide. Okay, let us take an example, carbon dioxide, and it can be converted into a supercritical fluid. And how do we do that? For this we have to reach a particular temperature and pressure, and at that point only this particular compound, that is carbon dioxide, will exist as a supercritical fluid. So whenever we are doing supercritical fluid chromatography, we need to maintain a particular temperature and a particular pressure. So in case of carbon dioxide, we have to reach a temperature point of 31.1 degrees Celsius and a particular pressure. So based on that, it will be staying as a supercritical fluid, and if we are not able to maintain a constant temperature and a constant pressure throughout the process, then it will not work as a supercritical fluid. So this is about supercritical fluid chromatography, and the mechanism is just the combination of this gas chromatography and liquid chromatography. So it will have basically the traits of both gas and liquid. That is, this fluid will be actually having a density which is less than the liquid but more than the gas, and it will diffuse faster just like the gas and less like liquid. So as a result, it is having properties of both gas and liquid. So this is about the supercritical fluid chromatography. And gas chromatography is very simple. Gas is the one which is used as a mobile phase, and liquid chromatography, we are using liquid as a mobile phase. Now coming to the next classification, that is based on the stationary phase which is being used. So here we will take the example of gas chromatography, and we will divide gas chromatography based on the stationary phase which is used in gas chromatography. First, if the stationary phase is a solid, then it is known as gas solid chromatography. If the stationary phase is a liquid, then it is known as gas liquid chromatography. And if the stationary phase is totally bound to the support, I have already mentioned that is a support which is on which we are coating the stationary phase. And suppose if there is a chemical interaction between the support and the stationary phase and they are bound together, then it is called as bonded phase. So in that case, we will call it as a bonded phase chromatography. So here you can see, suppose this is the support, and over it we have just coated the stationary phase. So in that case, we won't say it as a bonded phase chromatography. But suppose if the stationary phase is actually linked to the support, in that case we will say it as a bonded phase gas chromatography. Now coming to the classification based on the different types of samples interaction with the stationary phase, which is the major principle of chromatography, that is differential interaction between the stationary phase and the mobile phase. So here we are taking the examples of liquid chromatography. So liquid chromatography can be divided into all these headings, all these types, based on the type of interaction of the sample with the stationary phase. First is adsorption chromatography. So adsorption chromatography is basically the adsorption of the liquid to the stationary phase. Then we have partition chromatography, that is the molecules of the sample, that is the sample which is containing different components, will be partitioned in a particular layer of the stationary phase, which is used in liquid chromatography. It can be divided into three different types. We have normal phase chromatography, then we have reverse phase chromatography. Details I will be explaining in the upcoming slide. So in normal phase chromatography, we are using a polar stationary phase, whereas in case of the reverse phase chromatography, we are using a non-polar stationary phase, and the mobile phase will be just the opposite. Then we have hydrophilic interaction liquid chromatography, so which is just a variation of this partition chromatography, nothing much to explain about it. And then we have ion exchange chromatography, that is in which the stationary phase is basically formed of several charges, so it will attract the opposite charges from the sample. Then we have size exclusion chromatography, in which we are having different pores in the support, and as a result it will be selecting the molecules based on their size. Then we have affinity chromatography, in which we are having several biological compounds which are present on the stationary phase, and this will be binding to their specific complementary proteins. That is, if we have antibodies which are acting as a stationary phase, it will actually catch hold of the antigens. So this is used in that case. So these are the different types of liquid chromatography. Details I will be explaining in the upcoming slides. Now let's go to the next classification, that is based on the pressure which is applied to move the mobile phase. So here also we are having two different methods, that is the different types of liquid chromatography. So far the column chromatography which I have discussed. Suppose in column chromatography we can have both the gas chromatography, we can also have the liquid chromatography. So if the liquid is traveling based on the gravity, then it is just a simple liquid chromatography. But suppose if we are applying pressure to this mobile phase, that is the liquid, to travel, then it is called as high performance liquid chromatography, which is also known as high pressure liquid chromatography. And here a pressure of about 6,000 pounds per square inch is being applied to the mobile phase, and it is more efficient than the normal liquid chromatography. And then we have even more efficient form, that is UHPLC, that is ultra high performance liquid chromatography or ultra high pressure liquid chromatography. So here we are applying a pressure of about 15,000 pounds per square inch, and we also have the difference between this because both of these are column chromatography and liquid chromatographic method. So here we are having a column which is having an internal diameter, that is, as you can see, like suppose this is the column. So this internal diameter in case of HPLC is 4.6 millimeter, and the length of the column is 250 millimeter. Whereas in case of UHPLC, the internal diameter is about 2.1 millimeter and the length is 100 millimeter. So this is the length here, this is also the length here, this is the internal diameter, this is the internal diameter. So as you can see, the dimensions are less for ultra high performance liquid chromatography, and this is the reason why the pressure is more in ultra high performance liquid chromatography, and it is even more efficient than HPLC because here more pressure is applied to the mobile phase. Then coming to the classification based on the type of support. Okay, so support, it can be present in the form of column, as I have already mentioned here. This is in the form of column, or else we can have the support in the form of a plane. So under this category we will have different methods. First is the paper chromatography, which I have already explained, and another thing is TLC, that is thin layer chromatography, where we are using a glass plate and on that we will be coating some stationary phase. So here you can see it will be in the form of plane rather than being a column. So here the support is in the form of a plane, so it is called as planar chromatography, and if it is present in the form of column, then it is called as the column chromatography. This was the method which was used by Michael Sweet earlier for the first time for doing chromatography. And then we have an entity that is known as the multi-dimensional separation, that is basically here we are coupling chromatography with some other separation techniques, and as a result this will increase the efficiency of separation of the different components which are present in the particular analyte of the sample. Now let's come to the different terminologies. So first one is chromatograph, which is very simple to understand. Basically it is the instrument which is used to do the chromatography. So in case of gas chromatography it will be called as gas chromatograph. In case of liquid chromatography it will be called as liquid chromatograph. Then the peak which I have shown you earlier, that is the two different components which were coming and they were forming the peaks, that is A and B. So this entire thing which we obtain after the detector detects the sample in the computer, so that is what we call as a chromatogram. So this graph is called as chromatogram. The next thing is retention time and retention volume. So for this I will be taking you to the next slide. As you can see here, this is the peak which is formed for the component A, and this is the peak which is formed for the component B. So as you can see, it takes some time for this component A to elute, that is from the chromatographic system and being detected on the detector. So that time is what we call as retention time, which is represented by TR1A, and for the compound B the retention time is represented by TR1B. Okay, so retention time is basically the time it takes for a component to stay in the chromatographic system and being detected for the first time in the detector. And retention volume is the volume which is being detected by this particular detector. So that is about retention time and retention volume. Then we have void time. Okay, so void time, we have void time, as you can see here, void time and void volume. So void time is basically the time which is taken by those components of the sample which are not at all interacting with the stationary phase. That is, as soon as you pour the mobile phase in, which is presenting the components within it, it will not at all interact with the stationary phase, and as a result it will be removed out of the chromatographic system very soon. And you can see it is represented by this small flicker here. This represents those components which are removed as soon as the mobile phase and the sample which is being combined together is injected into the chromatographic system. So the time taken for that is called as the void time. It is represented by Tm, and the volume which comes out is represented by Vm, that is the void volume. And what we actually considered later on is the adjusted retention time and the adjusted retention volume. So adjusted retention time is nothing but TR dash, so it will be TR minus TM, that is the retention time of a particular component minus the void time, and adjusted retention volume is actually VR dash, that is VR minus VM. that is retention volume for a component minus the void volume. Now coming to the next entity, that is the peak width. So what is peak width? So here you can see these are the two peaks which are formed based on the components which are detected by the detector of the chromatography system. And here the base length is what we call as the peak width, and we can also get the width at half height. Okay, so if it is the entire height of the peak, at half height if we take the width, that is called as half height width. So this width, if it is more for a peak, it means it is going to make it difficult for the two components to be separated. So we always desire the peaks which are having less peak width, and this will actually help us to get a better resolution of the peaks of different components. So I will be coming to resolution next. So this is about the peak width. Now coming to the next thing, that is called as the retention factor. So retention factor can be represented by the alphabet K, as you can see here. So this is called as retention factor. So what is retention factor? Retention factor is nothing but the adjusted retention time by the void time, that is TR minus TM by TM. So TR dash by TM. So this is the retention factor. So it is basically giving us the idea of the capability of the chromatographic system to separate the components. So the efficiency of a chromatographic system can be first obtained by the retention time and the retention volume, adjusted retention time and adjusted retention volume. Then we have the retention factor, and then following this we are having the selectivity factor, which is also known as the separation factor. Then we have other parameters also which will also detect the efficiency, that is the resolution. Then we have the height equivalent to a theoretical plate and the number of the theoretical plates. I will be discussing that. So we can also find this retention factor by the another formula, that is Kd into Vs by Vm. So Vm is the void volume and Vs is basically the volume which is representing the volume of the fluid over the stationary phase. So this ratio is called as the phase ratio, and Kd is called as the distribution equilibrium constant. That is, it is indicating the distribution of the particular components in a sample between the mobile phase and the stationary phase. So this is another method, but mostly I think this method is very simple because we know about the retention time, we know about the void time, and with this we can easily calculate the retention factor. Now coming to the next thing, that is selectivity factor or the separation factor. So it is basically the ratio of the retention factor of the two different components. And here what we have to do is we have to put the retention factor of the compound which is eluting later. As you can see, it is B here. So it will be Kb by Ka. So if the retention factors for two different components is same, okay, if the retention factors are same for two different components, then the value of alpha will be 1. So it means the two different components will not be seen as two different peaks, and it will be difficult to separate those two components by method of chromatography. So this is about the selectivity factor. Then we have N, that is known as the number of the theoretical plates which can be formed. So basically, suppose if we take an example of a column chromatography and we find there are some bands, let it be A, and then we have some bands here, that is, let it be B, mobile phase is being poured here. So these areas, these three areas, in these three areas actually the analytes are not attached to the stationary phase. So it is basically in equilibrium between the stationary and the mobile phase. So it is going along with the mobile phase because mobile phase is flowing. So in this case we can say the number, the plate number, that is N, can be equal to 3, because these are the areas where you can see there is no formation of the band of the analytes. So that is what we call as N, that is the total number of plates, that is the theoretical plates. So the size of this plate can vary, and we are calculating it based on a theoretical assumption. So basically this is a theoretical entity, a hypothetical quantity, that is N. So N is found out by TR, that is the retention time, by sigma the whole square. So sigma is basically the standard deviation for different baseline peak widths. So this is one formula for N, and the more is the number of N, more is the number of the theoretical plates, and as a result more will be the separation of the components. So this number of plates, actually this increase with the length of the column, because more is the length of the column, more will be the number of plates. That's why we need to have a even better measurement, and that is what we call as H, which is also known as HETP, that is height equivalent of a theoretical plate, which is nothing but the length of the total column by the number of plates, that is L by N. So it is an even better measurement for this. So we can use retention time, we can use retention factor, you can use selectivity factor or the separation factor, you can use the number of the theoretical plates, we can use the height equivalent of the theoretical plates for measuring the efficiency of the chromatographic system, and then we can also use RS, that is resolution. So the resolution means the capacity of the chromatographic system to separate two components and to see those as two different peaks, that is two different peaks, how the peaks are separated. So if we have broad peaks, it will coalesce with each other and it can lead to the formation of a single peak. And if we have narrow peaks, that is the peaks which are having narrow peak width, in that case baseline width, in that case those will be seen easily as separate peaks. So the formula of RS is TRB minus TRA by WBB, that is the breadth, that is basically the baseline width of one peak and the baseline width of the another peak divided by 2. So this is the formula for resolution. So we have discussed about the phase ratio, then we have discussed about the separation factor, then we have discussed about the plate number. Efficiency can be measured by all the different methods. Resolution we have discussed about the height equivalent of a theoretical plate, also known as the plate height. Then coming to band broadening, so basically band broadening occurs because of more interaction of the analytes with the stationary phase, and as a result there will be an increase in the baseline width of the peaks, which is nothing but band broadening. So it is basically a phenomenon, that is we are describing the increased baseline width of a peak by the term band broadening, and as a result this can lead to the coalitions of two peak and they will be seen as a single individual peak. Okay, now coming to the next thing, that is the peak height and the area height is basically the length of the peak, that is the height, and at half height, as I have already mentioned, we can measure the width also, and area of the peak. So basically this entire thing is the area under the peak. So this area of the peak is the representation of the amount of a particular component which is being detected by the detector of the chromatographic system. So far what we were seeing, the retention times are different for different components, and when we will compare it with the list which we are having of the standards, that is the standard list, we can easily find out this is that particular component. But to know what is the amount of that component in this particular mixture, we need to know the area, that is the peak area, that is the area under the peak. So that will give us the idea of the amount of the particular component which is present in that mixture. So that is about the peak area and the height. Now let me discuss about the resolution. You can see here with the example of the diagram. So here, as you can see here, as we move from above downwards, the resolution factor increases. That is, here you can see it is 0.5, then it is 1, then it is 1.5. So as you can see, when the resolution increases, that is it is increased to 1.5, you can see the two different components as two different peaks. But when it was less, that is 0.5, the two different components were coming so close to each other that it led to the formation of a single broadened peak. So we are not able to actually differentiate between the two components as individuals. So that's why RS value should be high. That's why in that case we will get two different peaks, and the peak should be narrow so as to get a better visualization of the different components. Here you can see the original chromatogram. Suppose if we increase the efficiency, the peaks are separated properly. So this is about the different resolution factors and how resolution actually affects the chromatographic result, the result which we obtain on a chromatographic system. Now coming to the next thing, that is peak width and half height peak width. So as you can see here, this is half height peak width, then we have the entire peak height, then we have the peak area, that is the area under the peak. So these are the terms which I have already discussed. Then this is the retention time. Then coming to the next two terms, that is internal calibration and external calibration. So internal calibration is basically a process in which we take a particular standard and we also find the chromatogram, that is we also develop the chromatogram for that particular standard before we do it for the sample. That is the case of external calibration. Whereas in case of internal calibration, what we do is, along with the sample, we take one internal standard and we do the analysis, and for the standard which we are normally using, with that also we will take that same internal standard and we will do the analysis, and as a result here you can see there is an increase in one peak that is representing the internal standard. This is done for the purpose of internal calibration. So this is internal calibration and this is external calibration. So only difference between these two is in internal calibration we are using another standard which is different from the standard which we are using already. So that is called as the internal standard. So this is about the calibration. So we have discussed about the external calibration. We have also discussed about the internal calibration. Now coming to the two next terms, that is the derivatization and the extraction. So derivatization is nothing but changing the quality of the stationary phase or the mobile phase. So suppose if we have a polar stationary phase, so in that case we can make it non-polar in nature, that is by replacing the polar groups which are present in the stationary phase, like we have the hydroxyl groups, then we have carboxylic acid groups, then we have some amino groups and all. So if we replace them from the carbon chains like C18 carbon chain, then C8 carbon chain, then that is what we call as derivatization, and as a result it changes the nature of that particular stationary phase, that is it converts the stationary phase from polar to non-polar. Then extraction means whenever we are getting a biological sample, in that we are having the different components, so we will try to first concentrate it more so that we can get the more amount and we can get a better result. That is nothing but the process of extraction. So derivatization is very important because it helps to convert a normal phase chromatography into a reverse phase chromatography. As I have already mentioned, in normal phase we are having polar stationary phase, whereas in case of reverse phase we need a non-polar stationary phase. So polar phase will be converted to the non-polar phase, that is non-polar stationary phase, with the help of this process called as derivatization. So these are the main terminologies related to the chromatography. The most important ones here are the retention time, retention volume. Following that you should know about the retention factor, then suppression factor, then band broadening, then plate number, height equivalent, and resolution. So the important ones which will actually indicate the efficiency is the retention time, then we have the retention factor, then we have the suppression factor, then we have the plate number, then we have the height equivalent of the theoretical plate, and finally resolution. Now coming to the different factors that affect the chromatographic efficiency. So as I've already mentioned those things, now let's actually discuss those in detail. First we have the column length, so because it is going to affect the number of the theoretical plates, as I've already mentioned, but not the plate height, because this will be actually L by N. So if the length increases, the number of plates also increases. So as a result, this will remain constant. Next, the particle size of the support. Suppose if the particle size is smaller, then it will give a larger surface area and hence it will have a better efficiency. Then uniformity in the size, shape and packing of the support. Then flow rate of the mobile phase and the linear velocity of the mobile phase, then the temperature at which the process is being done, the temperature of the entire system, rate of the solute diffusion, then the viscosity of the mobile phase, because it will be deciding the retention time, because how the mobile phase is flowing, that the flow rate of the mobile phase. Then initial injection volume and also the connecting system within the chromatographic system, like the connecting tubings, then we have the detectors, they will be also affecting the efficiency of the chromatography. Now coming to the first major category, that is known as the gas chromatography. So here you can see a pictorial demonstration of the gas chromatography. It has got the following components, that is carrier gas source, then we have the injection system, then we have the column which is present within the oven in which the temperature is being maintained, then we have the detector, and finally this system is being connected to the computer, which is also the control system. So the gas which we can use as the mobile phase includes hydrogen, then we have helium, then we have nitrogen. Okay, so hydrogen, helium, they can be used, and we can also use nitrogen. In case we are using hydrogen, we should be very careful about maintaining it because it is a highly inflammable gas. So standard precautions have to be taken while we are doing gas chromatography, and also proper ventilation should be provided. For different types of gases, we have different types of detectors which will be acting better. So there are several types of detector in case of gas chromatography, like we have the electron capture detector, then we have the mass spectrometer, then we have the flame ionization detectors. So there are different types of detectors. There is a separate slide for that, so I will be discussing that. Now this carrier gas source, so either it can be a reservoir of this gas or it can be a gas generator from where it is directly being entering into the oven. And then we have the injection system where we are going to put the sample, and then without interrupting the gas flow, that is without interrupting the mobile phase flow, much these samples are released. So it can be directly given in the form of a liquid which will be vaporized at the port, that is a port from where the particular components are coming out in the form of gas. That is, with the help of a temperature of about 50 degrees Celsius, the liquid components are converted into the gaseous phase, and as a result this gas phase will go through it, and then the separation occurs, and finally it is being eluted and it is being detected by the detector, and we will get the columns in the computer screen. So computer is responsible for maintaining the temperature of this oven, for maintaining the rate of the injection of the samples, for controlling the gas flow, that is the mobile phase flow, and for setting up the detector. So this is basically computerized. Here you can see the different types of columns which we can use in case of gas chromatography. First is the capillary column. So in capillary column, the entire lumen will be free, and basically we will be having the support and the stationary phase, which is actually going to be the inner wall of this column. So inner wall of the column can act as the support and the stationary phase both, or else inner wall can act as the or else the inner wall can act as just the support, and over that we can have the stationary phase. So it is further divided into these following types. First we have WCOT, that is wall coated open tubular gas chromatographic column. So here you can see the wall will be acting as the support, and over that we will coat the stationary phase. Next is support coated open tubular gas chromatography column, in which the inner wall of the column is not acting as the support. Over that we will be having the support, and over the support we will be having the stationary phase. Then next is PLOT, that is porous layer open tubular gas chromatography column, in which we will be having a porous layer which will act as the support and the stationary phase apart from the inner wall. And then this is the packed column chromatography, that is basically a column chromatography in which the entire lumen is being packed with several particles, and these particles will have the coating of the stationary phase over it, or these particles can itself act as the stationary phase and the support. So this is about the gas chromatography, and now let's move on to the different types of detectors which can be used in gas chromatography. So we have the flame ionization detectors. So the principle of operation for this is the production of the gas phase ions from the combustion of the organic compound which is being eluting out. Here you can see this is basically a pictorial representation of the gas chromatography system. Then we have other methods like electron capture detector. So here basically the capture of the electrons by the chemicals with electronegative groups occur, and that is the method which is being used here. Then we have the mass spectrometry, which is usually combined with the chromatography. Here also there is production of the gas phase ion, followed by separation and analysis of these ions based on their mass to charge ratio. So this word is very important, that is based on the mass to charge ratio. Next is thermal conductivity detector, that is it does the measurement of the change in thermal conductivity of the carrier gas as the compounds elute from the column. Then we have photoionization detector, that is measurement of the gas phase ions that are produced due to the chemical ionization with light. Then we have flame photometric detector, and we have infrared spectroscopy, that is infrared spectroscopy is based on the absorption of the infrared light by the different components which are eluting out of the column. So these are the different types of detectors under this. If you have to remember, you can remember this mass spectrometry, because mass spectrometry is nowadays very much used in combination with the chromatographic system. So you can remember this, and here the gas phase ions are being produced. You can also remember flame ionization detector, because there also gas phase ions are produced. So this is about gas chromatography. Now let's move on to the next one, that is the liquid chromatography. Here the mobile phase is liquid, and we have different methods, that is different types of liquid chromatography. First is the adsorption chromatography, as I have already said. So here the separation is based on the adsorption of the chemicals to the surface of the support. So as you can see here, these are the chemicals which are getting adsorbed to the surface of the support, and some of the chemicals which are not getting absorbed, they are going along with the mobile phase. Then we have partition chromatography. So partition chromatography, here the separation is based on the partitioning of the chemicals into the layer of the stationary phase. This is the layer of the stationary phase, and these are the molecules which are getting partitioned in this layer of the stationary phase, and these molecules are going out. That is, these molecules are going more along with the mobile phase. Then we have ion exchange chromatography. So here there is the separation of the ions based on their binding to the fixed charges which are present on the support. So here you can see on the support we are having the negative charges, so it will be basically attracting the positive charges and they are getting bound to the stationary phase. And on the other hand, the negatively charged ions, that is negatively charged analytes which are present in the sample, will be going along with the mobile phase and will not be actually getting adhered to the stationary phase. Then we have size exclusion chromatography. So here basically we have different size pores in the support. So here you can see these are the molecules which are having the size which can be easily accommodated in these pores. So they are getting bound to the stationary phase, whereas these molecules are having a size which is more than the size of the pore. So they will be going along with the mobile phase. Then we have affinity chromatography. So it is based on the separation of the chemicals. That is, separation of chemicals is based on their interaction with the biologically related binding agent. So as you can see, we are having the binding agent. So it can be antibody, it can be antigen. So the molecules which are having a specific affinity for this particular binding agent will bind to it, and the rest of the molecules will go along with the mobile phase downwards. This image is of the size exclusion chromatography, which is actually a type of column chromatography. As you can see here, the small compounds are getting trapped in the pore, whereas the large compounds are coming out, that is, they are coming out fast, eluting fast from the column chromatography because they move along with the mobile phase. So this is in whole about the different types of liquid chromatography: adsorption chromatography, then we have partition chromatography, then we have ion exchange chromatography, then we have size exclusion chromatography, then we have affinity chromatography. So ion exchange chromatography is mainly used for the separation of different charged molecules like amino acids. Then we have different ions which are present, and size exclusion is best for separating the larger macromolecules. Then ion exchange is also used for the DNA molecules, then DNA, because those are negatively charged. Then we have affinity chromatography, that is used for detecting some of the antigens by using the corresponding antibodies so as to find the antigens which are present in the particular biological sample. So these are basically the different types of liquid chromatography. And partition chromatography is further divided into two types, that is the reverse phase chromatography and the normal phase chromatography. In normal phase chromatography, as I've already mentioned, we have a polar stationary phase and a non-polar mobile phase. Whereas in case of reversed phase partition chromatography, we have a non-polar stationary phase and a polar mobile phase. So normally the most commonly used type of partition chromatography is actually the reverse phase, because most of the compounds which we need to separate are actually ionic in nature. So for them, basically it is going to be better if it is having a polar sorbent, because in that case it will be easily dissolved and we can separate it based on the reverse chromatographic method. So here you can see a pictorial depiction of the affinity chromatography only. So the affinity ligand is basically the compound which is attached to the support with the help of a spacer arm, which is not having any interaction with the compounds to be detected. And this is to this affinity ligand that the molecules which we need to detect will be binding, as you can see here. And the molecules which are not having a binding tendency with this affinity ligand will be moving along with the mobile phase. And now you can see here we are having the workflow of liquid chromatography. So the different components which are present in liquid chromatography include: first is the solvent reservoir, which will be having the solvent, basically the mobile phase. Then we have the pump. Since it is a case of high performance liquid chromatography, we are having a pump which is going to pressurize and provide the pressure to this mobile phase. Then we have the injection valve or the injection system from which we are going to pour in the samples. Then we have the column here, okay, and this has to be maintained in a proper temperature, that is, a proper temperature has to be maintained. And in case of liquid chromatography, there is a special thing: we need to maintain the constant temperature throughout the process. That's why it is called as isothermal separation, okay. Whereas if the temperature changes with time, as in case of gas chromatography, then it is called as temperature programming. But we don't do temperature programming in case of liquid chromatography, and we also don't do it in case of SFC, as you remember, that is supercritical fluid chromatography, because a particular component can stay in the supercritical fluid state only at a particular temperature and pressure. Then we have the detector system, that is being the components which are being eluting out will be detected, and it is being connected to the computer which is producing the characteristic peaks which we want for the different components, and then we can compare it with the standards, and we will get the corresponding components which are present in the sample. Now, the different detectors which can be used in case of liquid chromatography includes absorbance detector. So, absorbance detector measures the absorbance of light at a given wavelength or a set of wavelengths by the components which are eluting out of the chromatographic system, that is the chromatograph. Then we have fluorescence detector, that is, it is detecting the ability of the chemicals to absorb and then re-emit light through fluorescence, that is re-emitting light through fluorescence. Then we have electrochemical detector, then we have conductivity detector. So electrochemical detector measures the current or charge as a result of the chemical oxidation or reduction of the components, and conductivity detector measures the changes in the conductivity of the mobile phase as the different ions elute from the column, that is different components elute out of the column. Then we have refractive index detector, it measures the change in the refractive index of the mobile phase as different compounds elute out of the column. And then we have mass spectrometry, that is based on the mass to charge ratio, the different components are separated. Then we have other methods like evaporative light scattering detector and charge aerosol detector. So these are the different detectors which we can use in case of liquid chromatography. And mass spectrometry, as you have noticed already, is used in combination with liquid chromatography, and it is also used in combination with gas chromatography also. So this is the depiction of HPLC, that is high performance liquid chromatography. As you can see here, we have the solvent, that is the mobile phase reservoir. Then we have the pump which is providing a pressure of about 6,000 pounds per square inch. Then it goes through this HPLC column. This can be of different types, and normally the columns which we are using are part of the reverse phase chromatography. That is, the columns will be made non-polar with the help of the process of derivatization, as I've already described it in the previous slides. So here we can have either C18, that is octadecyl, or we can have butyl group, or we can have octyl groups. And then it is being connected to the detector, which is being connected to the computer system, and the waste is being collected, and in the computer system we will get the characteristic peaks of different components. And here you can see the column which is being used. So it is having a coating of stainless steel outside. Following that, it is having polyether ether ketone, and then inside we are having the packing material. So basically this is a packed column chromatography method. It is not a capillary column. Okay, so this is about the HPLC. So HPLC is having an advantage that it is done very easily, it requires less time, then it is more efficient than the normal chromatography, whereas the normal chromatography is actually little bit qualitative in nature, whereas high performance liquid chromatography is more quantitative in nature. Then coming to the liquid chromatography, that is another variation of liquid chromatography, that is TLC, that is thin layer chromatography. As you can see here, we are having a glass over which we will be coating the stationary phase. So glass is actually acting as the support, and over that we will be coating the stationary phase. Then we will put this in this chamber which will be covered with the lid. And before putting this glass slide, what we have to do is we have to mark a line at the bottom of this glass slide here, over which we will put the drop of the sample which contains the different components, and then we will place it inside, and here we will have the solvent or the mobile phase which is just below the mark we have already made. And then with the help of the capillary action, the mobile phase will move upwards and the samples will be separated differently, as you can see here. And this is actually indicating the solvent front, that is the total distance which is actually covered by the mobile phase. And the entire process will be done in a controlled environment because we are going to put a lid over this chamber. So here what we are going to calculate is called as the retardation factor, which will be unique for every component. As I have already mentioned about the retention factor, so here retardation factor is basically the ratio of the distance which is traveled by that particular component but in the analyte by the distance which is traveled by the mobile phase, that is the solvent front here. So here you can see this is the distance traveled by that particular component ds, and this is df, that is the distance traveled by the solvent, that is the mobile phase. So this is about thin layer chromatography. And thin layer chromatography can be easily done here. The advantage is that we can actually see the different components as different bands and spots. The disadvantage is that sometimes these spots, when are having a size of more than 2 millimeter, then they can coalesce with each other and two different components cannot be seen as individual components. Sometimes it also happens that the solvent movement is erratic, and because of that the separation doesn't occur properly, and this will give us error in the results. And sometimes it also happens that instead of having one dot, the entire sample will be spread throughout the thin layer, that is throughout the thin layer chromatography support. So it will form a band instead of forming some particular spot. So that is a disadvantage of thin layer chromatography. And thin layer chromatography is more of a semi quantitative method, that is it is more commonly used in qualitative method, but since it is very easy to perform, it can be used in several laboratory techniques. So this is the thin layer chromatography machine, and you can see the different applications. It is used in pharmaceutical industry, it is used in material science, used in environmental science, forensic analysis, and also in life sciences. So I will be coming to the applications in the next slides. So before that, we will go to multidimensional separation, that is combining two different separation methods. So here the example which I am taking is liquid chromatography being combined with mass spectrometry. So this is the liquid chromatography system and this is the mass spectrometry. So when these two methods are combined, this will even enhance the separation even better. And first what it does is it does the separation in liquid chromatography, then the different ions are classified based on their mass to charge ratio in mass spectrometer. So it is possible only when the mobile phase which is used in a particular analytical method is compatible with the method which is used in the next analytical method, then only these two can be combined, and this will lead to the formation of multi-dimensional separation system. So this is about multi-dimensional separation system. Now coming to the applications of chromatography, first applications of gas chromatography. So it is used in food analysis, that is food analysis and quality control are one of the most common applications for gas chromatography. Accurate quantification of compounds and contaminants in food can be done, like the carbohydrates, proteins, vitamins, lipids, steroids, pesticides, and even some of the trace elements which are present in the food. Then next application is in environmental monitoring, that is with gas chromatography researchers can accurately determine the quality of the air and identify potentially harmful chemicals which are present in the air. Then it is also used in drug testing, that is sporting bodies like the Olympics and the professional leagues commonly use gas chromatography to test the athletes for the traces of the banned substance which they are using for increasing their athletic performance. Then it is also used in the forensic analysis. Then it is also used in manufacturing quality control measures, that is from automotive plants to pharmaceuticals. Many manufacturers use gas chromatography to test the quality and safety of their products. So it is used in pharmaceutical and the automotive plants industry also. Next, coming to the applications of the liquid chromatography. First is the application of the thin layer chromatography. It is used for identification of compounds like acids, alcohols, proteins, alkaloids, amines, antibiotics, and more. So basically the same application as seen in case of gas chromatography. Then it is also used for determination of the pesticides seen in gas chromatography and fungicides in drinking water. Then vitamins in soft drinks. It is used for rapid determination of group composition of heavy petroleum products, like we have asphalt, then we have pitches. For that we can use it. Then coming to the applications of HPLC. So first the pharmaceutical applications, that is it is used in tablet dissolution study, that is how much of the tablet is available for absorption, that is being calculated with the help of this tablet dissolution study in pharmaceutical dosage of the particular pharmaceutical dosage form. Then it is also used for pharmaceutical quality control, as I have already mentioned in case of gas chromatography. It is used for identification of active ingredients of a dosage form, used in environmental applications, that is just identifying the pollutants which are present in the water bodies. So that is the same application as we can see in case of gas chromatography. Then in case of forensics, it does the identification of anabolic steroids in different biological samples, that is in serum, in urine, in sweat, and in hair. It also does the forensic analysis of the textile dyes. It can also determine cocaine and benzodiazepine which is present in the body fluids of the individual who consume them. Then the clinical applications, that is it is used for estimation of bilirubin and biliverdin in blood plasma in case of hepatic disorders. It is used for the detection of thalassemia and other hemoglobinopathies. It is used for separation and identification of psychotropic drugs like antidepressants, benzodiazepines, and neuroleptics, that is the antipsychotic drugs which are present in an individual. Also this method is used for bioassay of different compounds, that is different drugs from the animal specimens, that is In case of chloramphenicol, it can be used in case of cotrimoxazole. In case of penicillin, for the bioassays of these drugs in different animals, we can also use HPLC. Then finally coming to the final application, that is in case of food and flavor, that is it is used for doing the sugar analysis in fruit juice. It is also used for doing the analysis of the presence of the polycyclic compounds in vegetables, then determination of the preservatives in the food and whether the food is of apt quality or not, as it is seen in case of gas chromatography. So these are the applications on a whole for the gas chromatography and liquid chromatography. Under liquid chromatography, we have the thin layer chromatography and the high performance liquid chromatography and ultra high performance liquid chromatography. Finally, coming to the summary of this entire presentation. So chromatography is a technique which is used to separate and analyze mixtures of chemical substance. That is, we are having a sample which comprises of several components and we are separating it and we are analyzing it. So it involves passing a mixture of this component which is dissolved in a mobile phase. This is very important. And this mobile phase passes through a stationary phase, and based on the differential interaction between the mobile phase and the stationary phase, it allows the component to travel at different rates, and this causes the separation. So this method is widely applied in case of research, as you can see, in drug designing, in case of pharmaceutical industry, in case of environmental testing, to identify, quantify and to purify compounds. That is, purify the compounds, then to identify a particular compound and to do the quantification, that is the finding out the amount of the particular component present in that particular sample. So this is about chromatography. These are the references which I have referred to for preparing the slides. So if you like my content, please do like, share, subscribe, comment, and also press the bell icon so as to get notified whenever I upload a new video. So thank you guys.